Hi, my name is Rudolfo Karl, and I'm a PhD student at the Institute of Innate Immunity, where I work on the interactions between non-immune cells and immune cells. And I'm the first author of the publication Renal Tissue-residient macrophages promote cystogenesis in early polycystic kidney disease. And in the next 2 minutes, I'm gonna show you what's all about. So my paper is about the polycystic kidney disease, and specifically it's about tissue-resident macrophages in the disease context. So over time we have an increase in tissue volume and tissue weight of the kidney, and here I'm looking into the interaction between tissue-resident macrophages on the one side and the renal epithelial cells on the other side. So for me, the most important or the coolest aspect of my paper is really like that we understood how the tissue-resident macrophage population behaves at steady state and during disease. At steady state, we partnered up with Elvira Maas from the LIMES, and we employed the double fate mapper model to see and understand that tissue-resident macrophages, especially during early stages, are mostly derived from the embryonic yolk sac. And then when we move towards the disease, we see this expansion of this tissue-resident macrophage population even before the onset of cystogenesis. So for me, the most striking part is really that we could understand the role and the behavior of the tissue-resident macrophages during slowly progressive and early PKD. So because what we could see here is that the renal epithelial cells, where the disease originates and which are forming the cysts, are acting on the tissue-resident macrophages, and they seem to reprogram them. And then the reprogrammed tissue-resident macrophages act back on our renal epithelial cells and lead to a worse disease outcome, to increased cystogenesis. My favorite figure is figure number 4, because there I have these 3D co-cultures of tissue-resident macrophages and renal epithelial cells, which are cultured together in a matrix made out of matrigel. And this really was one of the hardest essays to make to work. It took me a lot of attempts, many fails, but in the end I managed to really sort out primary macrophages, put them in my 3D culture. And now I have these very nice figures made out of ICC images of cysts, and of a lot of nice graphs made for quantification. So it really comes together very nicely. Of course, the work is never really done. So what we are now doing is really looking at how these two cell populations are communicating with each other. Like, how do the renal epithelial cells reprogram the macrophages? Is it direct cell-cell interaction? Is it like signaling via paracrine factors? And then, of course, what is changing inside the macrophages? How are they getting remodeled or reprogrammed? Like, we're going to do RNA sequencing, and we're going to do a lot of cyclotomics and proteomics, which we're going to do together with the group of Professor Felix Meissner, who is also like us at the Institute of Innate Immunity. So if you want to find out more about it, maybe check out our website. We are, as I said, at our institute here on the Venusberg at the UKB, or Universitätsklinikum of Bonn. And yes, if you want, you can check out the link in the description below.